So, welcome everybody to 2025 and the first in the year of 2025 of Hamlet BioPharma's in-house digital meetings where we make you more acquainted with the different projects that we are running and also with all the fantastic people who are involved in this work. I'm speaking English because today we have the great pleasure of having a visitor who's working closely with us normally, but not always present in Lund. Professor Farhan Haq, who is a specialist in bioinformatics. So most of the presentation today will be given by Farhan Haq. I would just like to start off the meeting by telling you a little bit about what's going on and where we are with everything. So just bear with me for 1 minute. And we will share the screen with you. So let's see. There. Where we have the full screen. Yes. There are always different things on different computers. So I hope now you can see the introductory screen that we use for Hamlet BioPharma's presentations. Combination of a hand and a test tube and a pipette and some beautiful imagery of cells, some of the cells that we work with, not in case from our lab, but a wonderful image. Så välkomna. Det första jag vill påminna er om när det gäller vad som har hänt sen sist i vår riktade nyemission. Totalt 15 privata investerare och family offices deltog i den emissionen som slutfördes strax före jul. Genom emissionen tillförs Hamlet BioPharma drygt 26 miljoner kronor. Och transaktionskostnaderna blev väldigt låga eftersom vi genomförde den här emissionen på egna meriter. Och jag vill framförallt tacka våra långsiktiga investerare som ställde upp och delar vår entusiasm och optimism när det gäller bolaget och som också var ett stöd i den här processen naturligtvis. Vi vill göra vårt allra bästa för att leva upp till förväntningarna och arbetar hårt med det. Första fokus är på våra 3 positiva kliniska studier som ni väl känner till: blåscancer, bladder pain syndrome och recurrent acute cystite. Och när det gäller blåscancer så har vi sedan ni hörde oss sist avslutat den longitudinella studie som vi har bedrivit under ett antal år i Prag tillsammans med det kliniska teamet där. Och att vi har avslutat studien innebär att vi har en första del som är den randomiserade placebokontrollerade studien med låg dos. Och en andra del som innebär att vi gjorde en doseskaleringsstudie med 5 och 10 gånger högre doser av alfa-1H. Och att vi nu har avslutat den kliniska delen av den tredje delen av studien där vi har ökat antalet patienter med 8,5, vilket är den dosen vi siktar på. Och där vi också har tittat på upprepad installation. Så när jag säger att studien är complete så innebär det att vi har gjort det som kallas för datalock, det vill säga att alla kliniska data och underlaget för klinisk statistik finns nu inlåsta hos det företaget som hjälper oss med studien, och vi kommer att genomföra all statistisk analys där tillsammans med dem. Det är också väldigt mycket patientmaterial som finns, alltså som vi måste matcha med kliniska data för att se hur det har gått för patienterna. Och också de molekylära analyser som Farhan Haq kommer att beskriva lite om vilka tekniker vi har för att utvärdera detta. Därmed så kan vi också göra en clinical study report. Det har vi inte genomfört tidigare för delstudierna, men nu har vi då material för att göra en hel studierapport. Och det viktiga är att detta är ju en del av vår dialog med FDA. Vi fick väldigt positivt svar från FDA i vår dialog med dem och med frågor som vi ska diskutera vidare. Väldigt glada över responsen och hur effektivt och snabbt de har svarat. Tillbaka till de andra delarna, det som har med infektion att göra, bladder pain syndrome och recurrent acute cystitis. Recurrent acute cystitis är ju, den studien är avslutad och där håller vi på med underlag för publicering, tittar på analyser av data och skriver materialet tillsammans med den tyska gruppen som ju genomförde den kliniska delen av studien. Och det är en väldigt positiv process eftersom alla är så glada över att resultaten blev så bra. När det gäller bladder pain syndrom fortsätter också studien som planerat efter den här initiala delen som vi har rapporterat. Vår filosofi när det gäller infektionsbekämpning är ju väldigt annorlunda och väldigt intressant. Och jag tar bara med några av de nya tankar och resultat som finns idag. Vi kommer att återkomma till det i framtida möten. Vår pipeline innehåller ju nya läkemedelskandidater för att behandla infektion och specifikt att också kunna behandla antibiotikaresistenta bakterier. Och det här är viktigt för att istället för att döda bakterierna som man gör med antibiotika och nya antibiotika och ständigt nya antibiotika, istället för att göra det så tar vi reda på vad det är hos patienten som behövs för att avdöda bakterierna, att patienten själv avdödar bakterierna. Och våra nya läkemedelskandidater visar väldigt kraftfull effekt i djurmodeller mot bakteriella infektioner. Det som också är viktigt att förstå är att det här är inte vaccination som man ska göra innan eller immunterapi som man gör på lång sikt. Utan det här är akut behandling med våra nya molekyler. Alltså en veckas behandling, samma typ av kinetik som man har med antibiotika. Och vi har också inget belägg för toxicitet i friska djur. Det här är viktigt. Vi kommer alltså att rapportera mera data under våren. Den 6 februari har Vetenskapsrådet en konferens om antibiotikaresistens där de har bett oss att delta. Och då kommer vi också att ta upp en del av tankarna kring kommersialisering av den här typen av substanser. And now I would like to switch and to introduce you Farhanak again, who will tell you about the advanced technologies that are available and his collaboration with us to achieve these goals. So please.
Thank you. Thank you very much. I feel pleasure to talk here, and it's honored that how Katrina introduced me. And first of all, I'll go through what we are doing here and why this next generation technologies, which are actually revolutionizing all the medical science and infection biology, and why it is important and what type of technologies we are using here in the lab. So first of all, before going into the detail, first we should know that finding the cause of the disease and finding the specific targets to those disease is very important. And for that, we need to understand what type of changes or what type of mutations or variations that are occurring within an individual, whether it's a patient of cancer or if it's a patient of any other disease. So if you see here, this is a slide presentation from a very famous scientist, Michael Stratton from Sanger Institute, where these sequencing technologies started. And he's if you see the slide, you can see that from the beginning, like for instance, from the fertilized egg to gestation, like even before the individual is born, at that time and throughout the lifespan from infancy, childhood, and adulthood, there are different changes that are occurring within the individual, and that can be either any change that can be a mutation, and that mutation can either be a passenger mutation or driver mutation. The difference between passenger and driver is that there are some changes which are occurring and they don't have a phenotypic change, but there are some changes which are very critical to understand because they actually change the phenotype of the individual and develop a disease. So, and those patients, those mutations or changes can further lead to any type of, for example, here's this example of the disease, cancer. So it will turn into a cancer, and that cancer can become invasive. And then there are, if we try to treat it with the conventional methods like chemotherapy, radiotherapy, then most of those changes are resistant to that. And before these technologies, we were unable to identify those critical changes that actually are resistant. So now what we are doing here is that, for instance, like if I tell you start from the very basic, that everything that is happening within the individual is either occurring at the DNA level, mRNA level, or at the protein level. And this is known as the famous scientist that described the structure of the DNA, that is Francis Crick. He said that it's the central dogma of the molecular biology, or you can say central dogma of the life. So all these changes which are occurring at DNA, mRNA, and protein, all of these are we are studying here, and we are identifying different features or changes that are actually related to the disease. So what technologies we are using here? That's a very famous term, that omics. That means omics is for either genomics or proteomics or transcriptomics. And in genomics, what we do is, as we know that I told you that DNA. So at the DNA level, we check whether the individual has any specific change in the sequence. So for that, I will tell you in the next slide that we use different technologies to identify those DNA level changes and how those changes are actually interacting between with the other changes to make to develop a phenotype that of a sick or a diseased or a cancer patient or an infection patient. And again, also we are working on proteomics, in which we are sequencing and identifying the expression or the levels of the proteins that actually are changing the phenotype of the disease. So we are also looking at the structures, how the structures uh, are changes, how the functions are changes, how different proteins are interacting with each other in those specific cases. And also at the RNA level, at the transcriptomic level, we are using different technologies to like microarray technologies and sequencing technologies to check the transcriptome of the genome, that which genes are expressing and what are actually associated with the specific uh, patient. And then, so what actually are those type of changes? If it's a very brief summary of all of them, like for example, as I shared that at the DNA level, and then the RNA level, and then the protein level. So at the DNA level, there are different type of changes which occur in the genome, like single nucleotide polymorphisms that can known as the point mutations, any specific change at the DNA level, or that can also be insertions, like multiple changes at the DNA level, or insertions, multiple new nucleotides are added in a specific gene or a specific region, or deletions, that are multiple nucleotides are deleted at the specific chromosomal or DNA or a genetic level. And then this CNV is actually the copy number variations. It's also a very interesting thing, that the whole chromosome, it's not only about the genes or the particular point. It comprises of number of genes within a one segment, which are actually differentially deleted or inserted. And then the structural variations, like how the DNA structure, like the gene fusions are occurring. So we check all of these in our lab, and we identified this, and I will show the examples later. And then after that, when we identify those changes at the DNA level, then we check that whether those DNA changes are actually turning into the RNA of the protein, that is actually the actual coding part of the gene, that is whether those mutations are uh, affecting the expression of the RNA of that particular gene, or by overexpressing it, like the oncogenes, or the underexpressing it, like the tumor suppressors, if they are regulated. And then we are also looking at whether those changes which started from the DNA turned into RNA, and then at the protein level, whether those are actually expressing in that particular disease. So next. So these are the basic, now if, from going from basics to a little bit complex, that these are the major technologies we are using here in our lab. And for instance, if you look at that whole genome sequencing, what we did here in different clinical trial studies and as infection studies, that we have sequenced the whole genome of the patients, and then we identified those mutations or copy number variations at the genomic level, at the exomic level, and also at the transcriptomic level, at the RNA level, that what genes are expressing and what are they doing. this is an abstract representation of how we detect those changes. Before 2000, there was nothing about the genome sequencing and these technologies. So when in 2000 there was a reference genome was identified, and then that reference genome is now available in public repositories. And what we do is that first we take any specific, for instance, we are talking about any cancer patient. We take the DNA from that cancer patient, then we take the RNA from that, we convert that RNA into the cDNA, and then we do sequencing of that sample, and that actually then we have a raw file, and that raw file, it contains everything about that patient, and now we want to know that this patient has what type of changes. So what we do is that we use these new generation technologies, these new AI methods and machine learning methods, that we compare that with the reference genome, and we look for these mutations that at what chromosome position, at what genomic position, or and what DNA or RNA or any specific gene these deletions, copy number variations, or the structural variations are present. And then we treat then we classify that and associate with the disease. So these are some excerpts from our very recent studies. We are actively working in a lot of different projects, as Katrina told you about. So this is one large-scale longitudinal study where we identified the DNA level changes and classified the infants which have a severe kidney infection with the normal babies, and we also did that in two independent populations. And what exciting thing was about it is that using these clustering methods, we classified that what are the biomarkers, you can say, or what are the genes or changes at DNA level which are occurring between these two groups, like what either they have a severe acute pyelonephritis infection, or if they don't have. So we have a list of biomarkers which actually are associated with this disease. And another study, what we also very recently published, and it's a phase 2 clinical trial of the bladder cancer. And what here we did was that we sequenced the transcriptome of all the patients, and then after sequencing it, we did the transcriptome analysis, and using different statistical and approaches, we identified that there are a number of genes, like out of all the 22,000 protein coding genes, the total number of the protein coding genes is between 22 to 25,000. So out of that, we identified a number of significantly less than 3,000 genes first, that which are significantly associated with the treatment of alpha-1 oleate in those patients. And then what we did was that we classified those changes, and we identified that the treatment of our alpha-1 oleate is specifically increasing the apoptosis or the cell death in the patients which have the tumor. So we identified the efficacy, that it's at the molecular level, that the drug is actually treating the patient. And also, this is the functional classification of those genes, that we identified that after treating it with alpha-1 oleate, these different type of disease genes which are associated with this disease are actually inhibiting the cancer invasiveness and metastasis. So yeah, so these are a few excerpts from our study and what we are doing here. And the last one is also, like I told you about the DNA, RNA, and then also at the proteomic level, we have also identified that how alpha-1 oleate is actually regulating the cytokines or the immune markers in bladder cancer patients. And we have found very interesting patterns of immune response associated with alpha-1 oleate in those specific patients. So that's a bit of a summary from what we are doing at the genomic level, and that's our team. And you can see in the center, Katrina. So Thank you very much.
Everybody is.
Yeah, yeah. Yes, everybody's on.
Yeah,
And if you have any questions, I'll happy to. Thank you very much.
that's great.
Thank you.
I know that this is technical, but we think it's an unusual treat for us to listen to you
Thank you.
because you can simplify
Yeah.
the message to a conceptual level. And I hope those of you who are not thinking about DNA and RNA every morning for breakfast.
Yeah.
There's a very famous scientist who also was awarded the Nobel Prize who said he used to read DNA sequences for breakfast. We haven't quite reached that level,
Oh,
but this was also some time ago. So we'd be happy to answer questions both about the company side and about this presentation. Maybe I could ask you. You're also an expert in identifying in computer programs that identify and predict biomarkers.
yeah.
And I know that you've commented on our studies that there are so many biomarkers.
Yeah.
How would you see that?
Yeah, like that's a very good question. Like for now, out of identifying, like for example, as I said, that there's 22,000 genes.
Yes.
And then from that, we using the sequencing technologies, we identified 2,000 something genes. And now here, what comes is that new technologies which we have published and we are using that. We use different machine learning approaches, artificial intelligence methods that from all those significant genes, what are actually the key markers
Yes.
that are actually regulating that are actually affected by the treatment
With
of
alpha-1 oleate or
So,
something else.
So there are a number of methods we are using, like multiple feature selection methods, machine learning methods, and then predicting the response of the disease.
And this is part of the data that we put together in the portfolio. Because one thing is if the tumor disappears or it shrinks in size, it gets smaller.
Yeah.
That's sort of the classical way of looking.
Yeah.
And then if you cut out the tumor, you can do pathology and look at the cells and the microscope and see if there are any tumor cells, and they are experts at that in Prague. But then you can also ask the question if the drug is changing the tumor in a more general way. And what you're saying with those 2,000 genes is that it showed that most of them were inhibited and
Yes.
cancer functions were inhibited and so on. So we can give a lot more detail, both about the mechanism and the response that was possible before this technical revolution.
Yes, the best thing about the best thing about the technology is that you can particularly identify the for instance, like the famous term personalized treatment.
Yes, yes.
Like you can identify based on the gene that how to treat a patient, and that's what we are.
Yes, and also things that are shared. We can say you showed us apoptosis.
Yeah, apoptosis.
So we see this response in all the patients that get alpha-1 and not in so many in the placebo group. So we also want to learn more about the response as a way of, it's part of the mechanism of action, isn't it, of the drug?
Yes, definitely, yeah.
It's fantastic to have this magnifying glass to look at the tissues. Are there any other questions from the audience?
Hi, Katarina. It's Timmy here.
Hey, Timmy.
Hey, quick question on that. So, like, how does this compare to other type of cancer drugs in terms of like how it impacts the genome and all those type of things? Like, do you see this in other cancer drugs? Or is this very specific for alpha 1H?
No, that's a very good question. That's very specific to alpha-1 oleate. And that's what we are doing at the genomic technologies, that definitely the other drugs have their own markers, or they are under process. But what best thing about alpha-1 oleate is that it's particularly targeting some specific genes which are specifically related to alpha-1 oleate. And we are filtering it down to the top key biomarkers, which we can use in future as a as a treatment for alpha-1 allele. Yeah.
If you think about cytotoxic drugs like what is normally given to cancer patients, I think they have quite a different profile. They don't trigger apoptosis, they trigger a nice form of cell death, but you don't know much more than I do about. You have looked at these profiles as well.
Yes, as Katrina said about the toxicity.
Yeah.
The best thing about this drug is also that it does not act toxic in normal patients, so it's only toxic for the tumor cells, and the apoptosis of the cell death is specific in the patients in the cancer patients which are you're given. Yeah.
There is one part which is common to other treatments, and that's Farhan. You're here somewhere. Yes, Farhan. Shut up.
Hello.
Hello.
Hi.
I do know you. Yeah, that's the proteomic analysis. So when urine samples were you screened all these patient samples, and then you saw that the response was very similar to BCG, right?
Yes, we didn't, for the first time when we analyzed all this data, we didn't know that. But when we checked the literature for more than 40 years, we saw similar overlap between this proton response in alpha-1 allele treated and also BCG treatment.
Yeah.
So it's a great finding for us.
Yeah.
We have different steps. We have influence or some effect in the RNA data, completely suppress the immune cancer
other genes,
gene, and then we have the protein response in this patient. So
that increases the immune
response. Increase the immune response to maybe remove the cell, remove the cancer cell, increase the cell shedding. Yeah. It's a magic molecule. I don't know exactly what happened there, but it's a magic.
It is. It's wonderful to see the effects in clinical samples.
Yeah.
You've done such a great job.
Thank you.
Yeah. Timmy, do you have any follow-up questions on this part?
Maybe. So, like, it's impacting like 2,000 different genes. Is that like any kind of comparison number to other thing, to BCG? Like, how many genes and stuff does that impact? Is there? Maybe.
No, in the BCG, the BCG studies are mainly related to a set of only 25 proteins, the cytokines. So basically, the BCG estimates, as Sharam can put light on it, that it's more of a immune response. They estimated only the immune response. They haven't done it at the transcriptomic or this big level. So we did both. We checked at the genomic level as well, and also we check at the proteomic level, and we compared it to the BCG because that's the standard till now. So that's why we saw that the proteomic response is also not generating any cytotoxic effect, and they are also enhancing the immunity. And the one good thing was that with alpha-1 allele, it is more rapid. As it happens quickly.
In a couple of hours.
And BCG takes weeks or months. I think you can.
Usually when they started to treat a patient with BCG, it's take a time more than 1 month to start the first line innate immunity. Exactly, to increase. And then they need to repeat it for 3 months and then continue it. But all these proteomics response happen in just 2 hours.
Yeah, it's very quick.
And then we have urine.
But we shouldn't generalize. I always worry, we shouldn't generalize about BCG. What we can say is that we are using a technology that has very high resolution. So we can say things in detail about our drug, and then we can compare. And it may be that other drugs have not been tested to the same extent with this in-depth technology. So it's hard to say they do or they don't. But where we have looked, we find these comparisons with BCG, and otherwise apoptosis is what everybody wants, right? That's the nice form of cell death, and it's the predominant cell death response in your, as you showed in your analysis. So this magnifying glass is working. I mean, there is also the potential to find new things, but we're focusing on alpha-1 very strongly right now. Do you have any other questions to us today? We will also, as I said, be present at Vetenskapsrådets meeting for antibiotic resistance on the 6th of February. And we have a time slot in Aktiespararna in their next meeting next week. We'll send out a message to all of you if you want to listen in there as well. Then thank you so much. We're happy that you took the time to join.
Thank you.
Thank you.
Take care.